Thank you. I want to first start by thanking the organizers for the opportunity to talk about the work that some of the people in the lab have been doing in the past few years. So this is really a story about diversity. So Sidney Brenner, when he was given the Nobel Prize, had this fantastic sentence, which is "Nature's gift to science." This was a joke with about the title of the main journals in science, but actually he was talking about this diversity of shapes, of forms, of sensory systems, of habitat, and nothing applies more than these concepts to mammals, and specifically to cetacea. They're really, they're really different. They have a number of characteristics that make them very unique, very special from many points of views, and we have been seeing that today in many of the talks that we've heard. So we have been trying to understand some of the origin, the genetic fingerprints of some of these morphological traits. And what sort of mix of bizarre traits do we see in cetacea? Some of them are rather visible. Their shape and form, of course. Some are not so visible. They have a very unique physiology, which is, in some cases make them a very special model to address, for example, human diseases. But from a morphological standpoint, some of these features are really unique. For example, when you look to the skin, and if you make, for example, an histological section, you won't see any sebaceous glands, for example. And that's very unique. I'll come back to that later on. But they don't have other aspects that you see in other mammals. For example, in males, you can see the scrotum. There's no hind limbs. They don't have olfactory bulbs. They don't have, for example, the elbow, this movement that most mammals can do, they can. So these are really very unique characteristics. So one has to ask the question of how do you make these unique phenotypes? Where do they come? And so in the lab, we've been trying to understand some of these traits by looking at genomes and making genome comparisons across a wide range of cetacean species. I'm going to give you 3 examples of how we can sort of get insights into the history of some of these traits. One is related with the skin. Another one is related with brain and behavior. A bit more, not so solid ground here. It's a difficult topic. And at the very end, I will explore something which is it's ongoing work relating cetacea and this novel concept of the Anthropocene, so the threats, the human threats that we are posing to this very special group of animals. So the skin. Now, when you look at some of these anatomical and histological features of cetacean skin, you immediately are confronted with a number of very special characteristics. For example, very rapid sloughing, almost no scab formation when you get injured. Hypodermis is really thick and fatty, we all know that. But perhaps the most visible features is the absence of hair. And if you look deeper into the dermis, you won't find any sebaceous glands or sweat glands. So what's going on here? So we start asking this question from a comparative point of view by looking into mammals and particularly into cetacea. So what you have here depicted is the general structure of a sebaceous gland with a hair follicle and the sebum production, a very important physiological component of your skin. And we start asking, what's going on? If there's no gland, what happens to the genes that are normally involved with sebaceous gland function? And what you have here is a very wide description of a number of genes that are critically and specifically involved in sebaceous gland function across mammals. So, for example, MC5R, if you delete this gene in mice, you won't get it. You won't get sebaceous gland. What you have here depicted is a number of enzymes that are related with lipid production in sebum in the sebaceous glands. So let me take you through this image, which looks a bit complex, but it's actually quite simple. What you have here is cetacean species in a phylogenetic tree. And all these symbols, they describe deleterious mutations in the coding sequence of these genes. For example, what you have here is a gene called AWAT1, is responsible for wax ester productions. And when you look to exon 2, there's a conserved stop codon in the middle of that exon. That means that this gene is not transcribed into a functional protein. So this gene is in fact like an old house. There's no one living there. It's a fossil. It's an eroded gene. And you see that time and time again in all these genes that we analyzed in the genomes of various cetacea, from Mysticeti to Odontoceti. And you see that also, for example, when you look at transcriptomic data sets, for example, from blue whale. So basically, cetacea are unique when it comes to the genetic fingerprint of sebaceous gland absence. And that correlates with the absence of many, many genes. And that extreme gene reduction, it's also related with many, many other phenotypic traits from the skin. Not only the pilosebaceous unit that I just referred, but also in genes related with inflammation and immunity, in the desquamation and keratinocyte differentiation. So it's like a minefield of lost genes when you look into the history of the cetacean skin. How about behavior? So most mammals exhibit something called circadian rhythmicity. This is mostly controlled, not uniquely, but it's also controlled by an organ called the pineal gland. The pineal gland produces an hormone which most people know. It's called melatonin, also known as the hormone of darkness. It's released into the blood during the night. It tends to control our activity, our rhythms. It's not the sole regulator, so to speak, but it's really important. Well, in cetacea, when you look from an anatomical point of view, what you see is something called a pineal regression, and it's paralleled by their unique way of sleep. When you look at the genes that are involved in the synthesis of melatonin, genes called ANAT and ASMT, and to the receptors that signal this hormone, what you found? Well, what you find is again the same pattern, is the presence of ORF interrupting mutations, mutations that do not allow the formation of functional proteins. So in effect, cetacea don't have a cycling of melatonin like we do, or mouse, or any other, or most mammals in effect. So they have these remnants of the genes in their genomes that we can study, but they have these regressive signs in terms of anatomy. The final example relates to lessons that we can take from studying genomes to try to sort of anticipate impacts in the Anthropocene, which is this idea that past genomic events can have an impact on the future, namely those related with the effects of pollution, of human-based pollution into the ocean. So a few years ago, Michael Hiller, in fact, some people in my lab as well, described the same pattern. So critical regulators of xenobiotic metabolism, in this case a couple of genes called CAR and PXR, which are nuclear receptors. They are sort of maestros of an orchestra trying to regulate xenobiotic response. Well, in cetacea, these genes are non-functional, okay? And this was put as evidence for the way that their diet is organized. We think this is not the entire history. So we started looking at this using a double knockout mouse model, so trying to emulate the conditions that we see in cetacea. And what we see is that genes that are critically regulated by PXR and CAR are elevated in the mouse. Moreover, and to conclude, what we have shown as well, and we have seen as well, is that gene families that are involved in xenobiotic metabolism are really contracted in cetacean species. When you compare, for example, cytochrome P450s, which are commonly known regulators of xenobiotic response, they are really contracted when you compare, for example, to hippopotamus or cow, which are the extant sister lineages of cetacea. So the take-home message really is that when you look at these fantastic animals, you see massive gene loss. And this hasn't been a process of simplification. This has been really a process of the adaptation. We see massive gene loss. We see that this is a response to an adaptation to a very specific lifestyle, to the life in water. And this has been done through a combination of various genetic and genomic processes, which I haven't really explored here, like co-elimination and hub disintegration. And finally, of course, I want to thank the people that actually did the work, Raul and Diogo and Raquel. So thank you so much for your attention.